Oncology Clinical Trials Types of Graphs. Introduction. In oncology clinical trials, a variety of graphs are used to present data in a clear and interpretable manner. These graphs help to visualize complex datasets, making it easier to understand treatment effects, patient outcomes, and other important aspects of the study. Kaplan-Meier curves. Purpose: to display survival data showing the proportion of patients surviving over time. Description: these curves provide a visual representation of survival probabilities over time, often used to compare the survival rates between different treatment groups. Example: a Kaplan-Meier curve showing overall survival (OS) or progression-free survival (PFS). Forest plots. Purpose: to present the results of subgroup analysis or meta-analysis. Description: forest plots show the estimated effect sizes, for example, hazard ratios and their confidence intervals for different subgroups or studies. Example: a forest plot displaying the hazard ratios for OS, that is overall survival, across different patient subgroups, which is age, gender, and tumor type. Waterfall plots. Purpose: to illustrate individual patient responses to treatment. Description: these plots show the change in tumor size for each patient, allowing for a visual assessment of the range of responses from complete response to progressive disease. Example: a waterfall plot showing the percentage change in tumor size from baseline for each patient. Spider plots. Purpose: to show the change in tumor size over time for individual patients. Description: each line represents a single patient's tumor size changes over time, highlighting the variability in response among patients. Example: a spider plot depicting the tumor size trajectories for patients in a trial over multiple assessment periods. Swimmer plots. Purpose: to visualize the duration of treatment and response for individual patients. Description: each bar represents a patient's time on treatment with markers indicating start, duration, and type of response. Example: a swimmer plot showing time to response, duration of response, and time to progression for patients. Bar charts. Purpose: to compare categorical data or summarize numerical data. Description: bar charts display data in rectangular bars with lengths proportional to the values they represent. Example: a bar chart showing the frequency of different adverse events or the distribution of tumor types. Histograms. Purpose: to show the distribution of a continuous variable. Description: histograms display the distribution of numerical data by showing the number of data points that fall within specified ranges, that is bins. Example: a histogram illustrating the distribution of patient ages or baseline tumor sizes. Box plots. Box plot and whisker plots. Purpose: to depict the distribution of a continuous variable. Description: box plots show the median, quartiles, and potential outliers in the data, providing a summary of the variability and central tendency. Example: a box plot comparing the distribution of changes in biomarker levels between different treatment groups. Scatter plots. Purpose: to explore relationships between two continuous variables. Description: scatter plots display data points representing the values of two variables for each patient, often with a trend line. Example: a scatter plot showing the relationship between drug dosage and tumor shrinkage. Line graphs. Purpose: to track changes over time. Description: line graphs connect data points with lines often used to display time series data. Example: a line graph showing mean tumor size reduction over time for different treatment arms. Pie charts. Purpose: to show the proportion of categorical data. Description: pie charts divide a circle into segments representing the proportion of each category. Example: a pie chart displaying the proportion of patients with different tumor types or response categories. Heat maps. Purpose: to display data in a matrix format where values are represented by colors. Description: heat maps use color gradients to represent data intensity, often used for high dimensional data. Example: a heat map showing the expression levels of multiple biomarkers across different patients. Conclusion: these graphs are essential for effectively communicating the results of oncology clinical trials, aiding in the interpretation of complex data, and supporting the decision-making process in clinical research and practice. Please like, share, and subscribe. Leave a feedback comment mentioning any topic that you want me to make a video. Looking forward for a positive response. Thanks for watching. Please like, share, and subscribe. Leave a feedback comment. Many thanks for your support and having great connection.